Hello Reefers, it is Felix, and this is the second episode of the Deep Dive podcast. After the overwhelmingly positive response of the first episode, today is all about coral spawning, specifically Acropora spawning on a commercial scale with Sam from SciReef as our special guest. We talk about how to get from corals in the ocean to spawning them in a lab to settling the larvae and growing them into mini colonies that in the end could find their way into your home tank. This episode is for reefers of all experience levels, whether you've witnessed coral spawning in your own tank or you're just curious to learn more about this milestone in the aquarium industry. Please leave all your comments and questions below. Sam and I will be reading through them and reply to as many as we can. And to celebrate SciReef's first tank spawn corals hitting the market, we will pick one winner from Germany to win a gift card to one of the only 4 stores currently selling the SciReef corals, as well as one winner from the US or EU to win a Tropic Marine Care package. To enter the giveaway, all you have to do is leave a thoughtful comment or question and let us know if you're from the US, EU, and or Germany. The winners will be announced on my Instagram and YouTube channel 2 weeks after upload of this podcast. You can find the rules and info in the description. The Deep Dive podcast is still pretty new. This is only the second episode, but I was personally super excited about this topic. So if you enjoy it, please share it and subscribe to the channel. I've already got ideas and guests for future episodes. But that's enough from me. Let's dive in.
First of all, thanks for having me. I'm very pleased to be chatting about corals and coral sex tonight. And so I'm Sam Nietzer. I'm a researcher originally at the University of Oldenburg. I've been working there in the working group of environmental biochemistry for now, it's almost 15 years. And since last year, I'm CEO of the SciReef company located here in the beautiful city of Wilhelmshaven in the north of Germany, where we produce corals through spawning for the commercial aquarium trade.
That's the really cool part, is it's not research only, it's also commercial. So at the end of the day, that's something that every reefer can benefit from.
Yeah, exactly. So as much as I enjoy doing research and being a scientist, but to go from research into the real life application and to provide something that hasn't been available yet to hobbyists, to people who really have this deep fascination and love for corals and reef tanks. So I think that's basically really a dream come true.
Oh, definitely. Yeah, I think a lot of people would agree. So you spawn corals. When and where have you started with like the coral spawning? When did that idea even come up?
Yeah, so the coral work has, like in the scientific context, has started in 2010 on the island of Guam in the Western Pacific. Personally, I've been a hobby aquarist since elementary school, basically. The classic freshwater tank, initially with guppies and stuff, and then a lot more different freshwater tanks. And then with 15, 16, I had my first seawater tank. So I was always, in the hobby and also fascinated with coral. And then during the studies, so I studied biology and then marine. My wife and I had the opportunity to go to the island of Guam for our bachelor thesis. So it's a beautiful tropical island, very fascinating place. They have their own university with a beautiful marine bio institute, and that's where we then could do our practical work. So back then, the professor Peter Schub was director at the Marine Biology Institution. With him, we basically did our theses. So Marine was working with coral larvae, with brooded coral larvae, Leptastria purpurea, which is super interesting, weird little coral that releases fully developed planula basically every day. So you can collect the ready to settle larvae. And then that goal was to identify bacterial strains which induce the settlement of coral larvae. And that was basically the foundation or one of the integral parts of what we do today, actually. Back then, I was working with sponges to look at what kind of chemicals produce sponges, what kind of defensive compounds are in there, and test them against bacteria. So in order to both have enough different bacteria, we isolated bacterial strains from crustose coralline algae, right, that we all know and love from our reef tanks. The crustose coralline algae, or CCA, as they're also being called, is a preferred settlement substrate for coral larvae.
So that plays a big role in what you're doing today.
Yeah. So the algae, they have certain properties that the coral larvae really like, and that make them settle. So during 2010, we isolated about 100 strains of individual bacteria and had them in pure culture, and then tested them individually. And found one strain that was really active to make the coral larvae settle. So with that bacterial strain, we then continued to work, and we almost every summer we would go to Guam and be there for the mass spawning then from 2011 on. So on Guam, the mass spawning of the Acropora corals is usually in July or August, depending on when the date of the full moon is, because it's about roughly 5-6 days after full moon. So we would go there in time. We would collect colonies, mainly Acropora circulosa, which is more or less hyacinthus, and Acropora digitifera from the reef, bring them into the facilities there, flow-through facilities, like really nice facilities where you have like perfect reef water being pumped up from the ocean. You have the sun coming from above. The moon is there shining for you. So it's very good conditions to do experiments. So then we would have the coral spawn in the outside facilities at the institute and collect the eggs and sperm. We can go into the details, what exactly is being done and how to get the larvae. And with these larvae, then we did different experiments. So for these initial studies, then we were looking at sedimentation impact on baby corals, because they're really, really tiny, and sedimentation then is something they really don't like. And sedimentation, if it's caused by humans, like deforestation, and you have a lot of runoff during the rainy season, or there's dredging for hydro construction, like harbor facilities, and there's being a lot of dredging, and all the sediments are stirred up, and then they disperse and settle on the reef. And if that happens in the wrong time of the year, when you have all the baby corals there, then it can basically wipe out an entire generation. And that's what we were looking at. In order to do the experiments, we needed the coral larvae. So that was one of the studies, and then we kept going to Guam for other studies and experiments, like looking at heat resilience in juvenile corals, because they're super fascinating, because they're so different from the fully developed corals. So on the one hand, they're super tiny, and in the beginning, basically defenseless. But on the other hand, they're still fairly plastic, meaning they can adapt to the prevalent conditions, and then they can cope with conditions that the parents cannot, basically, especially in terms of temperature, because they can still form the symbiosis with the Zoosanthellae. And if they grow up under higher temperatures, then the symbioses that form with the Zoosanthellae and the bacteria are different from when they would grow up under cooler conditions. But once these symbioses are fully established, then they can't really change it anymore, right? But that's the big hope, is that if there's enough rejuvenation, meaning there's always enough baby corals coming into a somewhat healthy reef, then it's there's a chance that it has, a not too dim future, I would say. So that is really fascinating. And in order to do these experiments, we needed to be better or more efficient, to become more efficient in producing these coral larvae and the juvenile corals. And then basically the grow out is also a very tricky part of the whole thing. But over the years, we got more efficient. And then in 2015, I think, we were standing in front of one of the grow-out tanks, and there was about 30,000 baby acros of about I would say almost a cent size, 8 to 10 millimeters and diameter. So it was 30,000 of them that we needed for experiments. And I was asking Maureen if she was aware of what you could do with these corals if they were allowed to enter the market, what that would mean for the whole trade, for the hobby, what that change that would be for a new way of producing corals. She was like, no, she's more coming from the research side of things. So she's not, or hasn't been back then. She hasn't been a hobbyist. So, but from that moment on, basically the idea was born. That,
there it was.
We should. Someone should definitely do that. And we also have the aquarium facility, the research facility at the institute in Wilhelmshaven. That was set up in 2011, and then from 2012 on, I was taking care of that. So it's a 6,500 liters recirculating system. And in the beginning, in the initial years, we were running it with the North Sea water.
Oh, really?
Which is actually, it's not as bad as some of you might think. But it's also not great. Let's put it like that. So in order to use North Sea water, we would, at high tide, when you have some cleaner water coming along the coastline from out, farther out in the ocean, we would pump in water into big concrete tanks, would let all the sediment settle down, and then it would go in the inside facility through a big skimmer and UVC and ozone and all that. And then you would run an ICP, and sometimes it would all be fine. Sometimes you have like 80 microgram of zinc in it. Sometimes some other crazy metal concentrations. And you would dump everything and pump new water. And then sometimes you have a perfectly fine ICP, and you use the water. And then the corals, obviously they didn't really like what's in the water. And then most likely there was some organic contamination, which we can't, we don't have a grasp on. So it's always a bit hit and miss, right? If the inorganic part is all great, then you have still no guarantee that there is not any organic stuff in there that you don't want to have. And then from the moment on when Tropic Marine said, hey, how can we support you? Then we switched to artificial seawater and have been using the Pro Reef ever since.
That's a bit more reliable, something you can count on.
Yeah, exactly. You know what is in there, and I would say almost even more important, you know what's not in there. That was a big change then. And then we thought, hey, you know what? Jamie showed that it's possible, and I don't think I have to introduce him here. I guess everybody knows Jamie Craig, the absolute pioneer of the ex situ spawning. So he was the first one ever to manage to get them spawned to spawn acros like in a planned manner successfully.
Because until then, you've only spawned corals in Guam, right? Or was it still happening in your research facility? Not at that point yet.
No, we had no. We had once that like one of our the green slimers that was producing eggs, but that was random because every day is the same in that facility, or was the same. So there's no way of knowing if and when a coral is going to spawn. So unless you do the seasonality, you can't spawn corals in a targeted way. So we were working with brooding corals, meaning the ones that have the internal fertilization and release the fully developed planula or clonal planula like the Acropora. But the spawners, we have only been working on Guam with them. But was a good timing that Jamie launched his spin-off company in 2020, I think, the Coral Spawning Lab. that would put together systems that have all the right components, so you can simulate the seasonality and get corals to spawn in ex situ environment. So that was a very good timing. So we thought, hey, let's get some of these systems. But our institute is also a bit like the coral reef. Like space is a very highly valued commodity. So we thought, hey, why not put everything into a shipping container? That's what we ended up doing. So Jamie and his team, they assembled the systems into an actual 20-foot shipping container, and then that shipping container made its way across the North Sea. And in that case, also tropic marine was also integral for us to obtain the container. So yeah, we're also very thankful for that. And then in November of 2020, the container landed in the garden, and we started the systems very quickly. And in December, then we had the first spawning. So we got corals from Australia that were still in the natural cycle. So they were exported in, I think, mid-November, late November. So when they arrived, basically in our systems, they arrived back in Australia. So it was a bit, stressful to cycle the systems in not even 3 weeks to get acros in, but that worked out. And then, we actually had the first spawning then in December of 2020.
Oh, that must have been something.
Yeah, that definitely was something. No, that was amazing. Yeah, to see that we have seen it so many times in Guam, but seeing it then in Wilhelmshaven in an artificial environment, that was a beautiful moment for sure.
Well, sounds definitely like something you've worked hard to achieve. So already at that point.
That was quite a long way. A lot of loops and paperwork and a lot of effort too. So, and over the years, we could identify a individual compound. It's like a red pigment. It's called cycloproteosine. And it's has a nice coloration actually. It's pretty red. And that is one of the inducers that make coral larvae settle. And now, since we know what kind of compound it is and we can produce it, then we have the ability to use a single compound or an extract to settle coral on a target substrate, like a clean ceramics or a clean plastic-based substrate. But you can avoid a lot of the hassles you have when you use a natural biofilm, like CCA covered rock, because then you have all the other competing organisms. And then it's one thing to settle 10 larvae in a petri dish and 10,000 in a larger container. So that upscaling is a lot of research work. So in that first research project, we laid the foundation to do all the processes in an artificial environment, spawning, or to get the corals spawning reliably and to produce the larvae, to get a feel for the species, because even though most acros, they reproduce in the same manner and the basic processes are the same, there's still quite a lot of differences in terms of how can you handle the larvae, how long do they take to develop, and how rough can you stir them, and and so on. So they're all a bit different. So you have to get a feel for the species. That's why you sometimes need a few runs to get to know how to handle the species. And then by the year, when is that, 2023, we felt like we slowly go into the aim at the commercial application. But still there was or is still a lot of research in terms of upscaling to be done. So yeah, that's where we are now. And we launched in March of 2024. And now in November 25 is when we start to enter the market with our first...
Made in Germany Acropora.
Yeah, exactly.
Awesome. That was quite the history. That was quite the way to get to this point. Like, there's a lot more behind this than you would think at first, honestly.
Yeah, it's been a wild ride.
Oh yeah, definitely. Well, I prepared a little background why I'm interested in the topic of coral spawning, which is not nearly going to be as interesting. Essentially what happened is I've kept a reef tank forever. One night, I just happened to look in the tank. One of my Acropora was spawning. It was Acropora Zoosanthellae, the Joker Acropora.
Awesome. Yeah.
And I was so excited. I was like freaking out. I was like, oh my God, that's like the best thing you can ever achieve in your tank, is like your corals are doing so well. They're spawning.
Well fed and happy.
Yeah, exactly.
Yeah.
And it's like all the stars literally aligned. And then I was like freaking out. I collected all the larvae. And then, it turns out you can't really do anything with it. It's just larvae of one Acropora in a bucket, and that's about it. So nothing happened. And I was kind of like, first I was so excited, and then it was followed by that disappointment of, that's it. Nothing's going to work out for you. So there's nothing you can do with it.
If it's just one colony spawning, then you have eggs and sperm from one colony, and that is a pretty frustrating thing because they can't really self-fertilize.
Exactly.
So maybe sometimes a few percent, but usually it's not
even then. What's going to happen? I mean, I'm not set up for it. I've just got this one tank here, and that's it. Whatever.
And also, if you have all the eggs that are not going to make it, let's say 1% is fertilized, then you have 99% of eggs that are going to dissolve within about 10 hours. And that's unfortunate reality of just one coral is spawning.
Yeah.
But of course, it's a great sign that the coral is doing well.
It kind of led me down this like obsession. I was a bit obsessed with the topic. I was like super interested. Yet it's this abstract topic that you can't really do much as just a hobbyist with your tank at home. There's not much you can do in that regard. But then it was last year at Interzoo, like May of 2024, where I've just attended Interzoo for the first time ever, and I walked past the Tropic Marine booth, and there you were with your little show tank of all tank spawned Acropora, and with all the little tiny perfect mini colonies. And then you had some bigger ones, the hybrid Nyos and Echinata, I believe it was.
Hybrid. We still don't really know what it is. We have we believe it's potentially hybrids, but we're still waiting for genetic data.
Well, whatever it was awesome. And you had
still here behind me in the room.
Do you?
Yep.
And I think there was one chimera in there, like a Nyos that was like green on one side and a bit of a creamy beige color on the other. Really cool. So that was like, I was like, okay, so you can actually spawn them and make them grow up into something in a tank. Like all essentially in a home setup, or well, not home setup, a bit more complex than that.
You
but
could, I mean, not in one tank, but in several. I mean, you have to. We can go into that. So it's highly unlikely that if it spawns in the tank, even if multiple colonies spawn in a tank, that they will reach the stage of actually settling. But technically, you could. I mean, it's a very complex process. There's so many individual steps, but
I know it's too complex, I think, for the average reefer to even pursue, honestly, and that's why it makes what you do even more special and amazing. Because I just wanted to understand, how do you get from a coral to it spawning to yet another coral, and now I get to talk to you and ask you all the questions. So that's awesome.
Yeah. Well, thanks so much. I remember really well when we were standing in front of the tank and talking about the corals and the spawning. And it's pretty complex processes, and each of the processes needs, I would say you can break down in also a range of complex processes. Some are more, some are less, but overall, it's nothing that can be easily done. I mean, especially not if you haven't worked with it for a certain amount of time, because a lot of things, I would say, even if we would write down a detailed protocol, it would still be
it's not something you just easily
you would still
replicate.
Need a few goals to get to find the to get grooved into how it's going. So I don't know. Should we go through the individual steps?
Yeah, let's follow the journey of a coral from selecting broodstock to finally a coral that can end up in your home tank. I think you touched upon the broodstock selection coming from Australia briefly. I think you mentioned that. So where do you get your broodstock from? Which corals do you select? Whether they come from? And why do you choose them?
So we have we work with Acropora because one kind of simple reason is we've been working with them on Guam for so long, because they're really accessible there. They're easy to collect. Used to be a lot, or there's still a lot of the species that we work with. You know exactly when they spawn, and the handling of the eggs and embryos and larvae we have done so many times that we got kind of a feel for it. But still, within Acropora is still every species is a little bit different, but generally, we have the most experience with these species. That's also why we started with them. And then we chose the Australian corals because they're quite accessible here in Europe. So you can get them through the wholesalers and importers from Australia. And then from these selections, the tenuous is one that is quite often imported.
It's a very popular one.
Yeah, popular one. Millepora is a popular one. And you always need several different genotypes, so not the same, not pieces of the same large table, but different ones. And with the tenuous, it's kind of easy because they're so highly variable in coloration. So if I have a beige one and a green one and a blue one, then I know, okay, these are different genotypes. Right? If I have 3 brown Millepora, they may be different, they may be the same. So that's the more variable they are, the easier it is also to make sure that you have different genotypes. Millepora comes in very often. Microclados, also a very interesting species that we work with. So that's, accessibility is a huge factor. So Australia, and then also Fijian corals. We also have these like Humulus and the Fijian tenuous. Speaking of tenuous, Australia is not tenuous anymore.
Yeah.
They're called Kentai since 2023. So,
but then Fiji is the actual tenuous.
Yeah, exactly. I think Fiji and Tonga are they are still allowed to be called tenuous. Yeah, and then Indonesia is Bifaria, and then I think there is one up in Okinawa that is I don't remember, but I think they split up in 6. And for now, who knows how many more there are going to be. So Australian Kentai, Millepora, Microclados. We have some Loripes, and then we also have some of which I have no idea what it is. We have offspring of these. They are. It's really pretty colonies, but I can't really say what it is. So that also makes it kind of interesting.
Did the Sorry, did the name change of the different tenues, did that make a difference for your corals? Like, did that impact your project at all?
Not really. No, I would no. As long as you know where the parent colonies come from. No, I mean it becomes then interesting if we have Fijian tenuous. Although the Fijian tenuous may already be a mix, but let's say they're tenuous, and you put them in the Australian system, and then you have the Fijian actual tenuous and the Australian kentai, and they spawn at the same time. And if you're able to hybridize these, then it's becoming a bit tricky.
Yeah,
to with the nomenclature. But that's also why we try to always know what coral or I would say which parent pool created which coral, so we don't lose the overview over time. Yeah, so in the Fijian system we have Humulus quite a lot. We have Nasuta, which is a beautiful species. I think also growth shape-wise and coloration-wise. We have the Fijian tenuous. We have Acropora austera in almost all, no, except for the Saudi system. So we have austera in the Fijian and the Australian system. And that austera comes actually, so the actual mother of the babies, so is from the Natural History Museum in Karlsruhe.
Oh, really?
So we got frags for the research project from Hannes in 2022 and put them in the Australian system. And then we got smaller austera colonies from Fauna Marine, so from Claude, and also put them in the Australian system in spring of 22, and then in fall of 23 they spawned.
Oh, that's awesome. So that's
which is awesome.
Sorry?
Which is fascinating because that means you can basically take any coral, even if it's been in the same conditions for 10-20 years and has never has seen much seasonality, and then you put it in a seasonal system and it takes a while until it's clocked in. But if it's doing well, then it's going to spawn at its internal time, basically.
That's really cool. So that's something you've learned then, I guess, because I was actually wondering if any of your broodstock colonies came from not just the ocean, but like classic hobbyist strains that we know under a certain designer name or something. Or, well, I guess you already mentioned you've got some from a public aquarium. So they don't have to come from the ocean, which is also really cool.
Yeah. So, I thought that's fascinating as well. Yeah, I think Jamie also did years back experiments with like taking from one biogeographical region the corals and put it in a different system. And then they take a while to adjust to the new seasons. And then they, just follow their internal clock. And that gives you a lot of new opportunities to also collect particularly beautiful corals that are in the trade, and try to see what happens if you get them to spawn, and cross with another beautiful specimen of the same or similar species. So there's now a lot of different possibilities opening up.
Oh, yeah. My mind's already going way far ahead right now. So why, I just have to ask this because I think that's a question we should ask earlier on, is why did you choose Acropora? I mean, I guess you kind of answered it already, but from a commercial point of view, you chose Acropora, which they're not the easiest coral, for one thing, to keep in an aquarium. So why not choose something like, well, they're not easy to keep, but when you keep them, they grow fairly quickly and they're easy to frag and everything. So why, or have you ever considered also doing this with other corals like LPS?
Yeah, that's definitely the plan. But in order to get started, we had to do what we can do best. And doing it also with the acros comes, of course, they're kind of tricky usually, but it comes with the opportunity to show what a difference it makes whether the coral comes from the ocean or whether it's been born and raised in an aquarium from the first second of its life, because that is making a huge difference when it grows up under aquarium conditions. We can see that, especially when it's a species like Kentai and Tenuous that are kind of notoriously finicky, and often people don't really want to even get them.
Don't get me started.
Well, I'm going to throw it out anyways after a while. So then, if you can then see how much more forgiving the offspring is, then I think that's also a good way to show the benefits of that type of reproducing corals, because it really makes a difference. And
so would you say that the difficulty has come down from like Acropora being the crown jewel, like the hardest to keep coral, I would say, and it comes down like how far? Are we talking like LPS difficulty, or is it more like maybe like a Montipora, so still like tricky, but a bit more forgiving?
Yes, they're tricky, but in case of the Tenuous Kentai, I would say they go more towards the easier acros. And the other thing is, I really like the morphology. So it's like that bonsai effect. If they grow from a larva, they look like early on. Not all acros do that. So Millepora often grows like horizontally a lot before it starts to branch out. Microcladus does the same, but the Tenuous Kentai, they early on they form these really tiny, beautifully shaped colonies, and then it looks like an actual colony and not just a branch that was cut off. So I think that effect and the variability, especially in a species like Kentai, so yeah, it's amazing if you take a close look at them, and there's so much you notice. Like from afar, you see the different basic coloration, the variability in the juvenile corals, but the closer you get, the more variance you see in between them and how different they are. So that's really fascinating, because I mean they're all siblings or half siblings and all genetically unique animals. So they also all look a bit different. So I think that's very fascinating. But the plan is definitely to branch out into other genera, like Trachyphilia or Scolimia, especially now that the import or the supply chain situation is getting more and more restrictive, especially with the LPS now from Australia. So we were able to obtain some before the import stop took effect. So hopefully we'll be successful with these guys, because that would also be very fascinating.
Oh, for sure. But it's good to know. So that is not something you've overlooked. That is something definitely on your radar.
Yeah.
Okay. Yeah. And then back to Acropora. So did you notice, like, obviously there are some Acropora that grow a bit faster, like Milliporas, the Tenuous. And then there are some that grow a bit slower. You mentioned the deep water. Now I forgot the name. The no, not the Ripes.
Loripes I mentioned. We have these.
Which
this one,
like or like something like just what we would consider a deep water Acropora in the hobby that has longer, thinner branches with fewer polyps. So do you notice a difference between the ones that have like way more branches and polyps compared to the ones that have fewer branches and polyps? That those are the slower growing ones? Do they then also produce fewer offspring or fewer larvae?
It's a bit tricky. I wouldn't particularly say slower growing, because some of the weird looking offspring of the Fiji tenuis or mixed Echinata tenuis. They also have the really long corallites, and they grow also fairly fast. But now that you say it, in the Kentai, we also have differences in the morphology. So some of them have the more tubular, longish corallites, and others are more stocky and dense. And the ones with the longish corallites, they are growing a bit slower. Yeah, I would say so. Yeah.
So does that have an effect on the amount of, baby corals they produce in the end?
So I would say, yeah. So the compact colonies, like or species like Humulus, even Kentai, Austera also has very thick branches with large polyps, and the corallites go pretty deep into the skeleton, so there's plenty of space for eggs. So the eggs are then lined up inside the corallite, and if you have a thin branch or a thin corallite, I'm not saying that all corals with thin corallites would generally produce fewer eggs, because I don't have enough data on that.
Okay.
Let's put it like that. But like a well-fed Humulus or Austera, they can pump out a lot of eggs. Also, like Digitifera on Guam and Hyacinthus or Circulosa. That's I would say they have more room to produce a lot of eggs.
All right, then I guess we can move from. Now we have the broodstock, right? We have the broodstock, and now the challenge is, how do we get that broodstock to spawn now that you have it? So how do you go about that? Like, how do you set up your tanks? How do you cue the season, I guess? What goes into that? So where do you start?
So basically the underlying principle is that the coral needs to know what day it is. Right? Because they have to spawn on the same day. So all the corals in the system have to be aware of which day it is. And they do that by following the lunar cycle. So they have light receptors, and then they can recognize a light impulse. At full moon, for instance, in the entire night, you have some light impulse, and new moon, then it's dark. So basically with these, and then all the intermediates, but then like full moon, light all the time, and then new moon is dark. So this basic rhythm allows them to know what is like a month or less, or what day within a month it is. And then on top of that is the temperature curve, or temperature seasonality. So usually in some areas it's quite significant. So northern Great Barrier Reef, you can go from, in the winter, which is our July, we're at below 24 Celsius. And then in summer, which is now, it can go up to like 29 and above. So that's quite the range.
That is the range.
If you would simulate it 1-to-1. Or in case of Saudi Arabia, we have at the Institute, we have a Saudi Arabian system. We have, I think in winter is like 24-ish, 25. And in summertime, it can, if you take the field data, it can go up to 34, which is quite crazy. So we don't simulate it like the full span, but we go to like 30. So with that, you have the temperature curve and the lunar cycle, and then the length of day is another factor, because then in summertime the days are a bit longer than in wintertime. The closer you go to the equator, the smaller the difference gets, basically, until the difference is very small. But these three factors are sufficient to get them to spawn simultaneously. There's maybe something also to the intensity of irradiance, but I think temperature and lunar cycle are the most relevant ones.
And so if you mentioned that you can put any coral into your, well, system that has the seasons kind of programmed into your lights and your heater and whatnot? And how long does it take for any coral to adapt to that system?
Yeah, so basically the system conditions have to be suitable for the coral species, of course, but let's say we run a medium extreme system like Australia from like 24 to 28, 29. I think you can put quite a large variety of corals in. And I think it depends on the timing you put it in. So for us, it was about 1.5 years with the Austera. But it probably can also be done quicker if you, let's say you have a coral from a system that is running at 24 and you put it in when it's like, or let's say you have 25 and then you put it into an Australian system when it's approaching winter and you put it in at 25, then it goes to 24, a bit below, and then ramps up. And then that would be like 7 months. So maybe that would be the shortest I would assume is possible, because the egg production is about 6 months in many species of acro. So I think when they get that cold phase about half a year before the spawning, maybe that's when then the egg production is ramping up. But you also don't know whether the coral that comes from a well-running system, maybe it already has eggs. Like in your case, it has already been producing eggs.
It just happened to have eggs, yeah.
Yeah, maybe if these are even almost fully developed, then you could put it in like 2 weeks before the spawning date, and then I would assume it would then spawn at the right timing. But yeah, I would give it about a year, just to be safe, 1.5 years.
But then still, within a year, roughly, you could then get any coral large enough to spawn. So what's the limiting factor? Like when do you know a coral is ready to spawn? Is it like a certain size or is it a certain age?
Age for acros, it's about 3 years after settlement that they are fertile and can reproduce themselves, and then from then on, depends on the lifespan. That's also a very fascinating topic, because there's still very little, or not. I mean, there's some things are known, but it's a field that needs to be studied much more. So how long do corals live? Because some we know can grow to be really, really old. I think the Acropora palmata like 1 or 2 centuries. I mean, I don't know off the top of my head, but I think there are studies that showed they grow really old. And then other species are, not as long-lived. And then we don't know what is the age limit of a certain coral. There's also, like the more compact, the corymbo smaller species, I would say like a humulus, you see in the reef, like 10, 20, 30, maybe 40 centimeters. But not much bigger. I mean, maybe they reach that's their size and then they stop, and that's all good. But then, you come back the next year and then the large colony is gone. Right? So, and then we don't know, is it why did it vanish if there wasn't like some severe disturbance? So I think that's a really fascinating topic. And some like the Caribbean Favia. Big Favia fragum. It's a brooding coral, and they have a fairly short lifespan. So they, after settlement, I think they can reproduce by releasing larvae after like 9 months. And after 5 years, then there's not much time left. So then they start to fizzle out. So these are rather short-lived species. So the range from just a few years to like the huge varieties that can grow to probably thousands of years. It's a vast range. So,
it's definitely interesting. The age of a coral is something as a hobbyist you don't really think about. But I guess we do have some corals in the trade that are, I don't know, they've been in the trade since marine aquariums are a thing. So maybe like 25, 30 years or something.
Yeah, even longer. I think the Stüber Acro, the Berlin Stüber Acro is still around, and that was from the 90s, I think, or late 80s. So almost 40 years. But yeah, there's also varieties or species that then suddenly started dying off in different people's tanks, and at the same time, or within a certain time frame, they all started to die, and not because people were doing mistakes, but maybe because the
you don't even know.
Was reached. Yeah, so that's also topic that we should at least keep in mind for the future of the hobby.
I mean, I don't think that's gonna be something that is really a common cause. I think the fewest corals that don't make it in a reef tank die because they're too old. But I don't think. I think that's very far down on the priority list of things that can go wrong, and then it died from old age. Probably
exactly.
not.
No, at the moment. But if we assume the supply chain of imported corals will get more and more restrictive, and then the trade here has to rely basically exclusively on what is in the trade, and then I have a hard time believing that everything would be covered for the next 30, 40, 50 years without running into problems like that. So
I think that's a really interesting idea in general, because you can still frag a coral the common way as you would. But then is there like a timer on a certain strain, or does just making a frag reset that timer? Or do we eventually have to rely on a coral that is spawned and essentially starts from zero?
Yeah, I don't think there's a reset. Because also if we look at spawning ability, so if you take a frag from a adult coral, then it will also spawn the next year, basically, or the same, or whenever you take the frag, then it's still ripe enough or adult enough to reproduce.
Really?
So, then it doesn't need another 3 years to reproduce.
Huh. Well, so now we've got your lunar cycle, we've got temperature. We've got time of day or duration of the day. We've got an old coral. What else do we need to make them spawn? I remember you said something about nutrition. So do you feed them something specific?
Yeah, so if we imagine how much biomass they release during the spawning in terms of the eggs, that's pure condensed energy. Basically fat and protein and the sperm also. So that's a lot of high grade compounds that they need to produce and at some point obtain. So the better quality food they have, or the availability has to be given, then they can produce their maximum amount of eggs and sperm. So we do feed them. We give them a sort of a smoothie. Kind of like everything that lives in the ocean, and run it through the blender, more or less. No, it's based on different fish and shrimp and squid and mussels and algal concentrate, and also put in the particulate phosphate and some fatty acids, and blend it, and then I freeze it. I used to do that on Guam as well. Especially in the early phase when they're still little, and you feed them in addition, then they grow much faster. So I would always get the mixed seafood bag at the supermarket and run it through the blender, and that was the go-to for the Guam babies. But here we do basically the same. And important is to not overdo it. I think to not feed them, like, even if they react positively, like in the moment when you put the food in and the materials come out and they seem very happy, if you do that too often, I think you can run into like infection issues if there's too much organics around. So we usually feed them like 2-3 times a week.
Okay.
And ideally at night.
Ideally at night.
So
yeah, when they're open.
Yeah, exactly. At night they go into feeding mode. Then you have the axial polyps. They come out like as far as they can. I mean, during the day you can feed them as well, and they react. But at night they are already in feeding mode, and then are probably most efficient in taking the food up.
Yeah, that's
so. Yeah.
All right. And then what else do we need to make them spawn? Not that that's not enough, but that's already quite a lot to make sure is in check.
They aren't disturbed, like the. So either if you want, let's put it like other way around. So the spawning is usually at night after sunset, right? So for Kentai is about, I would say, an hour after sunset is when they get ready to spawn. So they need darkness, and it's about, let's say between 4 and 8, 9 days after full moon. So, and in the days after full moon, it's dark after sunset, right? Because then you have sunset. At full moon, you still have the moon the entire night, right? The next day, you have about, let's say, 40 minutes of darkness, and then the moon comes up, right? And then the next day, it's like 80. Did I say hours or minutes? Not.
It's fine.
I said hours. I meant minutes. So you have sunset, and then about 40 minutes darkness, and then moon comes up, and then the next day you have probably 80 minutes darkness, and then moon comes up, and so on. So the initial time after the sunset is more or less complete darkness, and you can then either, like the way we do here at SciReef, because we can't get the main room dark enough yet, we do the spawning at night, right? And then you have to be here at night as well. At the institute, in the container, we can block off the lights because we have like shutters, and then the container, of course, can be closed, and we have doors in between to block off the lights, so you can also work in darkness when it's like 2 p.m., which is very convenient. So
you would have your corals spawn in the daytime where we'd also be awake, and you wouldn't have to stay up late at night, like
exactly
the last few days you have to catch them spawning in the middle of the night.
Exactly. Yeah. So, but then you have to be able to really block out the light, because otherwise then you may disturb their internal rhythm. Because often it's also, especially in the ex situ system, that not all species or not all colonies go exactly at the same day, which would be amazing. Like on Guam, you have the Digitifera. You can basically they all go the same night, right? And when they get ready to spawn, then you can also switch on the light, because when they spawn, then there's no stopping anymore. But then there's also no risk of disturbing any other corals, because all of them spawn. And that's not the case here, because we have different species, and then within a species, not all colonies often go at the same day. So you want to make sure that everything's in the dark, and you operate with red light, because they can't recognize the red light. So by using red light, then they still think it's dark.
Yeah.
But
so
if you got that,
no, we're kind of already sliding into the next point, which would be the spawning night, right? And you already mentioned the light's a bit of an issue. So in your system where you have corals spawn on different days, now the trick is, because you can't just turn on the light, because that would then, your one coral would still spawn, but then the other ones would get disturbed by the light and maybe not spawn anymore the following days. So that's a bit tricky, and that's why you have the red light.
Yeah, exactly. That makes it
it's
quite tricky.
Yeah, it's funny, because that's the trick that works for crabs and shrimp. So if you want to see crabs and shrimp at night in your tank, you use a red light, because they can't see that too well or at all.
Yeah,
so that's funny.
Exactly. Yeah. Also with some fish, right? When like imports come in, then often importers unpack with like red light. freak them out. Oh yeah, no, it's fascinating.
Same
So
trick
we
works
use
for corals, yeah.
like red headlamps, and then you can collect. So the way it works with the spawning night is you check your corals depending on the time. Like for the Kentai, it's like hour after sunset, they would get ready to spawn. Sometimes a bit earlier even. So you shine the red light into the tank, and when they prepare to spawn, they do a so-called setting, meaning the polyps, they inflate, and you can see the bundles that consist of eggs and sperm are already in the inflated polyp. And in the red light, they reflect kind of whitish, so it looks a bit like a Christmas tree with all Christmas balls on it. And then you know, okay, that colony is gonna go tonight.
That's cool. So you can see, so you have a bit of a visual cue, but that's like a last minute. That's essentially when you see that, you already know it's gonna happen like tonight.
Yeah.
Or is that like in the following? No, that is when that happens.
No,
it's happening.
Yeah. So with the Kentai, you have probably like, I would say, between 45 minutes to an hour before it releases the bundles. For Millepora, it's a bit longer. It's different from species to species. So some species go really late and do a long setting phase, and some species go late and do a short setting phase. So it's a bit different. But usually you can catch a queue beforehand. And then what we do is we put a transparent, like an acrylic cylinder, we place above the coral, and the cylinder goes above the surface. So once the colony releases the egg bundles, then they drift up to the surface because they consist of to a high percentage of just fat and have high buoyancy. And once they are in the surface, they're still in the cylinder, so you know all these bundles are from that one colony, and then you can collect them out with like pipettes and collect them in little plastic cups. And then you have all your plastic cups from the different colonies. And then it's on to fertilization.
So when you collect the eggs, you turn like the flow off and everything as well? Or do you leave the flow on? Because, I mean, in nature, there'd be like waves even at night. It's a bit calmer, but you'd still, everything would mix right away. So you kind of you don't want that to happen in that moment yet. Or is that something you do on purpose?
No, usually I would turn off the flow pumps.
So you don't want the eggs to mix yet, even if it's the same species of Acropora?
Yeah, but I want to do the fertilization. I want to get them all out of the tank, because once they refloat around the tank, then it's very hard to get them out of there. And if they stay in the tank, even if they're fertilized, they have a very little chance of making it, because the time frame from fertilization to a ready to settle larva is about a week. And I mean, there's so many predators, like the other corals at night. If there's eggs from other species floating around, of course they eat them. That's great food. And you got all the other critters and the pumps and the skimmer and the UVC. And it's a very hostile environment for pelagic little organisms.
Very fragile.
So, yeah. So we want to get as many of them, ideally all of them, out of the tank. And as I mentioned, the eggs and sperm are packed in bundles. So the acros are hermaphrodites. They produce both eggs and sperm, each polyp, but they can't self-fertilize. Even though they have sperm and eggs, then there's nothing going to happen. So you always need two. And these bundles usually consist of, depending on species, between 5 and 15 eggs. That are about 1 millimeter, maybe a little bit smaller, so it's also species specific, and then thousands of sperm cells, and all that is being held together by a lipid membrane, right? And so it floats up to the surface, and then you can collect it, And depending on species and temperature, this membrane is gonna dissolve, and then the eggs and sperm are separated. So then you can mechanically separate the eggs and sperm and then do the fertilizations.
All right. How do you move on to then fertilize them? So is there some controlled, like, do you know up front what corals you wanna, or what eggs and sperm you wanna mix, or are you like still in a trial and error phase, I guess, or?
Yeah, a bit of both. So it always depends on how many colonies of one species go. If you have just 2 colonies go, then it's kind of obvious. The path going forward. If there's more, then you have the ability to do targeted crossings, and then you have to bring together the eggs and sperm. Have to be in the right concentration. So if you use too much sperm, it's not good, and if you do too little, the fertilization rate may not be good. You leave them. Yeah, it depends also on for a certain time you leave it together, and then you have to remove the sperm as good as possible, or as good as possible, because the sperm cells also start to die off after a while. And also, again, depending on species, so then you transfer the fertilized eggs into either like chrysal containers or bowls or whatever you use to want to use to rear them. We use like a mesh bottom floating chrysals with a horizontal flow. What was it? After, for the Kentai, it's pretty quickly, so it's like an hour. Then you can see the egg cells starting to divide. So then they become peanut shaped and go into 2-cell stage, and then 4-cell, and so on. And then you know, how well the fertilization has worked. So ideally, you have a really high fertilization, because then you have you're gonna have more larvae, and B, you have not as many eggs that are dying and dissolving, because the eggs that are not fertilized, they will start to dissolve after like 10 hours, and I mean they consist almost exclusively of fat, and that fat gives then a fat layer on the surface, and then the other embryos can stick to and form clumps, and then the first one starts dying, and so on. It's like a domino effect. So the more...
Probably one of the few things that I can say, I know what you're talking about, because that's exactly what happened when my Acropora spawned. I collected all the bundles, and then didn't know what to do with them, and the next day it was like an empty bucket, and I was like, where'd they all go?
They're
just
that
dissolved.
whitish sludge then. Like a fat layer on the surface. So that's the worst case. But ideally, then they have high fertilization rate, and then they go through certain embryo stages. So the next morning, usually they're in the so-called cornflake stage.
I remember that from London as well, from your talk there. The cornflake.
Like cornflakes. It's pretty funny. And they're also super fragile at that stage. So if you, look at them the wrong way, they may fall apart. And then you have the whole thing of dissolving dead embryos again. So that's a bit a finicky time frame. But once they go through that, and then they become more roundish, then they're a bit more sturdy. And then after, I would say, also again, depending on species, like after 3 days, they're round. Or sometimes 2 days, and then they start spinning around their own axis. On the microscope, you can see that the little orbs are starting to spin, and then they start to develop the cilia, the little hair with which they can swim around in the water column. And the further they develop, then they become more elongated, like pear-shaped, and they are not on the surface exclusively anymore. So when they develop, they use up a lot of the fatty acids, and then they lose buoyancy, and then they go deeper into the water column, and then they start to swim around. And then by the time they are exploring the bottom of the container, then they're getting ready to settle. And then it's the time to take them out of the embryo container and start the settlement process.
So we kind of went from the eggs and sperm have now turned into like a larvae. And now it's kind of time for the larvae to choose its final destination, its final home. So how do we go about that? How does a larvae choose where to settle down? Because it's quite the important decision, I guess, for a larvae for a larvae to make.
Yeah, it is a very crucial decision, because usually where they settle is where they spend the rest of their lives, right? So if it's a great place, then it can be a very long life, and if the place is not that ideal, then it can be over in a very short time. So they have to be able to kind of recognize or identify the surfaces. And as we talked about earlier, they really like in nature the crustose corallines. And the way they identify these and the bacterial community that is living there and is probably the indicator for the larva that there is CCA growing, they have chemoreceptors. So meaning they can recognize chemical compounds, or basically smell. So they can smell their surroundings, and you can really see how they also in the containers, when they're ready to settle, they swim along the bottom and go from one spot to the next, to the next, to the next. So you can really see it trying to check out what
picky
kind of surface is there.
Yeah, you wouldn't expect anything else from an Acropora than being picky.
Yeah, exactly. Very good point. Yeah, and then you transfer these larvae into the settlement containers, depending on what you want to do. We usually have boxes, and then the process itself is a bit tricky. So they have to be exposed to the compound, but not too much and not too long, and with a right amount of light. And if that all is suitable to their needs, as you rightfully stated, they're acros, can be a bit picky, then they attach to the substrate, and they like certain topography, or like little nooks and crevices that they can nestle into. And then they attach and go through a metamorphosis into a primary polyp. So that goes fairly fast, actually, within a few hours.
Wait, before we get into that, so the larvae, when it chooses its spot to settle, I'm thinking it can't change its decision later. So it needs to choose at this early point in life. It needs to choose a spot that is suitable for a large colony. So I'm thinking high light and high flow. But wouldn't that be a very tricky spot to find for a larvae, especially the high flow part? So how would you go about that? Because of course you're not doing this in the ocean. It's in a controlled environment. But do you still have to provide it some amount of flow and light that makes it think, this is a great spot. Here I can grow into a big colony?
No, it's more about the chemical signals, and then I mentioned topography. So because they're really small and more or less helpless, they like to settle also in nature in like little cracks in the rocks, so they don't necessarily settle on top of the CCA surface, but in like a little bit underneath or in the little cracks, so where it's a bit more protected, and then grows from there. They don't. To my experience, they don't prefer higher light intensities to settle on, but rather like have areas that are a bit more protected.
All right. So no issues in that regard. So in your controlled environment, it doesn't really matter if they get blasted with flow, like a big colony would be.
Yeah. So we're not using a lot of flow. Maybe they could, because you don't. We also want to have them kind of evenly distributed on a certain surface. Right, and if you incorporate a lot of flow, then you would have probably a more patchy distribution. But technically, they can attach under high flow as well. If you think about the Opus larvae that sometimes in the tank just pop up on the flow pump or in some crazy spots. So technically, they have the ability even in high flow to attach.
But you want to attach, you want to enable as many of them to settle as possible for your goal is to grow, I guess, as many as possible.
As many of the larvae, but also not too closely together. Because, they can fuse. That's also a super interesting topic we should cover. But, if there's like 100 right next to each other and they fuse, then okay, for a few days they form these huge colonies where you think, oh wow, that's going to be amazing, but these never last.
That's a
so
pity.
If it's too many, so if it's like two or three, sometimes a few more can fuse and then form these chimeric colonies, but if it's too many, then they're not going to make it.
Then once they've settled, how long does it take for them to turn into a polyp? I think that's where you left off.
Yeah, so it can go fairly fast to like a primary polyp, like the flower shape, that can be a few hours.
Oh, really?
So once that process has started, so that they attach and then they kind of fold in, and then you see the septae structures forming, and if everything goes well, then most of the acros, they start to calcify after about 2 days. And then you can see the white, the skeleton structures start to form. And not too long after that, they are able to take up not necessarily food, but the Zoosanthellae. Because by the time they settle, they don't contain any symbiotic algae.
That's one point that I was super interested about, because where does the Zoosanthellae come from?
That's a super interesting topic also in terms of thermal adaptation. So in nature, they settle, and you have a high diversity of free-floating symbionts in the water. So the Zoosanthellae, they can live as like pelagic phytoplankton, if you will. They really like to be symbionts in colony or in corals, but the corals also maintain a certain symbiont density, right? So that means they constantly, if they're doing well, they excrete Zoosanthellae, meaning also in an aquarium tank, there is Zoosanthellae flying around or swimming around that are but the mother colonies are being released. But in an aquarium system, you have often like UVC, you have ozone sometimes, you have skimmers. So again, it's a pretty hostile environment for the pelagic organisms. So you don't have that many symbionts in the water. And that was for a long time was quite the bottleneck. On Guam, we have ocean water that is quite full of all sorts of symbionts, and also in nature, the symbionts or the Zoosanthellae, they don't, even if they're pelagic, they don't like to swim all the time. So often they sink down, And then on in the shallow areas of the reef where you have like sand patches, on the surface of the sand patch, there's often a really high density of the free swimming Zoosanthellae that just lie around and enjoy the sun. So if you scoop those up, then you can you have a lot of Zoosanthellae that you can provide to baby corals then. But that's not possible here. So if they don't take up symbionts, you can feed them. You can keep them alive for 4, 5, 6 weeks, but at some point, if they can't take up symbionts, then it's over. At least in my experience. Maybe if you put all effort and a lot of research in, maybe you can grow a post-symbiotic acro. That would be pretty interesting. But if they can't take up this Zoosanthellae, then you'll have quite some mortality.
I mean, it is a symbiosis. They need each other to survive. So, but it's interesting because then you have a coral without Zoosanthellae, still able to live for a while, just like the Zoosanthellae can be pelagic or sit around somewhere for a while. Yeah. But essentially, at the end of the day, they still need each other.
Yeah, Zoosanthellae, they're not as dependent, so they can. Yeah, they're fine. I mean, they like to be in a coral, but they're not. It's not essential for them, and that's also very good for us because you can culture.
Oh, so you do culture them?
Yeah, and then you can offer the Zoosanthellae.
Are there? I mean, now that I think of it, are there different types of Zoosanthellae, or is it all one?
No, there is more than 100 described strains, or some of the strains are now species. But I would say there's a variety of more than 100 different known ones, and they used to be grouped in one genus, Symbiodinium, and then into clades, clade A through G, I think, A, B, C, D, E, F, G. And now they have been, it's also has been 10 years almost, I think. They have been revised, and the individual clades now became their own genera. Right? So clade D, for instance, which is known to be heat tolerant or has a lot of heat tolerant strains, is now called Durostinium. And clade C, which is one of the most common clades, especially in the Indo-Pacific, is now called Cladocopium. And there's then different types of Cladocopium, and they're all a bit different. And also in terms of what they can provide to the coral. So what is really interesting, what we did in a long-term experiment, we exposed the freshly settled acros to high temperatures or to different temperature regimes and gave them a high diversity of different symbionts and then followed them over 1 year. Genetically, also what kind of symbionts do they take up and what do they have after a year. And we had them, the experiment started back then on Guam. The lowest temperature was 29, which is the summer temperature of Guam. And then we had 30 Celsius and 31, like constantly. And at the end of 1 year, we could see that there was not really a difference in survival. They were all fine. But the hot ones were much smaller. They volume-wise, I think they were like a quarter of the volume of the 29 Celsius one. And they all had pretty much exclusively one strain of Durostinium in the high temperature, whereas 29 had mostly Cladocopium and also different ones. So that was super interesting, and the 30 was kind of intermediate.
So that what can you learn from that? Because I'm thinking you end up with a more heat resistant Zoosanthellae. But if that's the only one that survives at temperature, that gives the coral less energy. Is that why it's grown less?
Yeah, it's more costly for the coral to survive at the high temperatures. However, the parent colonies, they would die within 3 weeks at the 31 Celsius. Right, so that was quite impressive that the juveniles, like the new generation, was able to survive that. And I mean, they were smaller, but they're still, doing sort of fine. But on the other hand, it's more costly for them. So that the Turistinium are more demanding, and they don't give as much to the host, and they kind of take a lot. But still, it's sort of working out, and you have a much lower diversity of symbionts, right? So if then one issue comes in that affects that symbiont, then they all have a problem again. So it's kind of the two sides of a medal. But it gives some hope that the new generation has the ability and the plasticity to cope with conditions that the parents could not. But once that symbiosis is established, right, and the interactions between the coral cell and the algal cell are kind of fine-tuned, then the symbiont cannot be replaced with a different strain. So that's the big problem that the corals have. So some corals have two types. Usually they have just one, whatever the type it is, but they have one type or maybe two, and then they can up and down regulate the quantities. So let's say it gets hot, then they upregulate the D, but they cannot, when they bleach and expel the symbionts, they cannot replace them with a different one, because that's what people thought in like the 90s when research was going more into the bleaching. And researchers thought the bleaching is like a fantastic mechanism for the coral to just throw out the algae that don't work and take some that are better suited. But that's not shown to be the case, unfortunately.
Yeah, probably not. Yeah, you just but that's still interesting, because you do see corals that completely bleach out and they're white, but that doesn't necessarily mean they're dead. Like, they still have polyps, they still have tissue. It's not like the bare skeleton, it's just the color is gone, the Zoosanthellae is gone, but they can repopulate with Zoosanthellae. Like,
they can recover, so sometimes they keep a very small percentage of the symbiont still in the tissue, and that can then repopulate, or they can take up the exact same strain from the water, and then through cell recognition they know, okay, that's the right symbiont, and then that can repopulate.
So just a thought, it's kind of like, could you then just synthesize or breed the Zoosanthellae that's like more heat resistant in like a larger quantity, and like just dump loads of it in like a coral reef where the corals just got bleached from a heat wave or something?
No, you would need the juvenile corals that have the ability to take these up.
Okay.
If the bleached one originally had different ones, then they can't really.
So that's what you meant. They can't. A large colony can't take new ones in. It has to happen at an early stage.
Yeah, it's like a fine-tuned engine where you can't just take the one screw and replace it with one of a different size or something like that.
That would be too easy.
Yeah, exactly. Then we wouldn't see the wide scale losses through bleaching that we do. But yeah, on the other hand, it gives a little bit of hope to use these methods of reproduction for restoration purposes, and then kind of future-proof the corals that are outplanted.
Are your corals ever going back to the ocean, or are your corals strictly for the aquarium market then?
No, I don't think it would be legally possible, and it wouldn't make sense logistically or ecologically to produce them here and then fly them to the global south and outplant them. But what we want to do, and that has always been, so for now we're focusing commercially on the aquarium trade, but in the midterm vision is, of course, that the refined methods that we're developing, we could also, of course, apply in the countries of origin, right? And then support projects with these methods to produce corals for restoration purposes. So from the beginning now on, we also support a project in Mafia Island in Tanzania, Eastern Africa. It's called Ropes of Hope. And they're doing really cool work where they collect corals that are broken off either by like destructive fishing techniques or like weather events, and they collect these fragments and then let them heal in nurseries and then plant them out on the reef again. And they're doing fantastic work. We've known them for a long time. One of the researchers there is doing her PhD and now working group at the institute. We've gotten quite some insight into the project, and it's super cool. And they also want to incorporate then sexual reproduction into the concept. So for now it's fragmentation, and then at some point they want to do both. And that's also where we, of course, want to go and support projects like that.
Awesome. Right, so I'm just thinking of the journey of the coral again.
We drifted on quite substantially.
Yeah, a little bit. But now we've essentially gotten our coral larvae settled. We've now got Zoosanthellae in it. So it's now essentially, it can eat, it can gain energy from light thanks to the Zoosanthellae. Now we gotta grow it out, right? So, I mean, at this point, how different is it from growing any Acropora?
Up until, I would say, 3 months, it's still quite different because they're still very, very small. They're very vulnerable to competition, through, let's say, other algae that grow, or maybe if you then have the CCA starting to grow on the substrates. They're tiny and helpless and tasty. Basically, a perfect snack for almost everyone. So if it's like amphipods or the Acerina starfish, that they are like the Death Star. One Acerina can eat 10, 15 little corals a night. So there's a lot of competition. You have to make sure it's not happening. So we keep them in separate tanks, of course, but basically the tanks are running as a normal system. They're also susceptible, especially in the first weeks, when there's too much organic or dissolved organics in the water, that you can get some bacterial outbreak that can wipe out quite a lot because they're so small and fragile. But once you reach like 3 months ish, and they are of a bigger size by then, it depends also on the species. They can be like slowly approaching 5 to 10 millimeters.
That was after how long?
Like 3 months.
3 months for a centimeter or a half inch or.
That's fairly big at that point, but I would say like a centimeter. They can in diameter. They're still then fairly flat, or the canthi are starting to branch. Then basically that's when the first branches occur. But from then on, they're more similar to a regular coral. And then it's also
a centimeter. That is like the tiniest frag you've ever found for sale anywhere. There's probably you can hardly call that a frag, to be honest, but
yeah. And then that's also about the size where the actual color is slowly starting to emerge. So before, they are just brown from the Zoosanthellae, and that's also exactly what you want to have, more or less. But then sometimes when we had people visiting, we had baby corals in that stage, like tiny brown things, little tiny brown polyps. We're like, that's all Acropora kenta. Okay, it looks a bit like Aptasia.
Oh no!
Wait. Be patient. And then after like 3, 4, 5 months is when then the first colors are appearing, and then from then on it's goes fairly fast that they grow to. Now at 11 months, the biggest ones are like 5 centimeters.
Oh wow! So there's quite a bit of growth then. Like early on, they seem to grow quite fast. Like it takes them a while to reach that, but I guess growth is still exponential.
Yeah, I mean they start off very, very small, and then they have a lot of processes internally that have to develop. But once they reach like that, roughly centimeter, then it's really a growth spurt. And we even had to kind of slow them down a bit because we have them on the grow-out plates, and all our corals are then being transferred onto our own plugs. And these plugs we had specifically designed, so they're made of PVC, and one gadget is that you can unscrew the stalk, then glue it into the reef, or you glue the stalk in, and then you can take the coral or detach the coral if you want to place it somewhere else, or move it to a different tank, and so on. And these came in August, and by that time it was already getting very crowded on the grow-out plates. So we even reduced feeding a bit, reduced light, so they don't grow too fast, and it gets too it gets too
they don't grow into each other.
Yeah, exactly. Yeah. They don't all grow wildly into each other. So
or start stinging each other, even worse.
Yes. So now we're 11 months after settlement, and the biggest ones are like 5 centimeters, which is quite a substantial coral.
Yeah, that is a mini colony, that is.
I think we can reach, for the Ken type, like marketable size of like 3 centimeters, which is also I would say a decently sized coral. That would be the size of the plug is 28 millimeters, I think, in diameter. So it would be about the size of the plug, and that should be possible after 6 months. If
that's quite speedy, yeah.
Yeah.
And then especially because you don't have your single branch, you have your little perfect mini colony. Looks like a mini colony too.
Yeah. It's like the bonsai effect. And even if you have a small tank, then you can have it looks like a reef with nicely shaped coral colonies.
It's like a real reef, but scaled down.
Yeah.
Another question. Before, when they're still small, do you use sea urchins to keep like algae at bay, or have you?
No, not at the moment. No. Jamie published a really cool paper about that, and it's a really cool method, but we haven't done it yet. So I would like to, but it's also time-wise, it's at the moment not possible.
But now that I brought it up, just to explain it, so the idea is to spawn sea urchins, and then have the juvenile tiny sea urchins eat all the algae that would compete with the settled corals in like the early stages.
Also the coralline algae, and the baby urchins, they don't do harm to the baby corals, so that's a very good combo. Especially the Mespilia, I think, is the best one for that.
That is the tuxedo, the blue one.
I think so. Mespilia globulus.
Yeah, that's the tuxedo, exactly.
Yeah.
I wonder how they found that out. Out of all the critters in the ocean, they found that sea urchins don't eat the larvae, but everything else. Like, why not a snail?
On Guam, we have the really tiny hermit crabs, the Clibanarius humilis. They grow to be like they're really small, and in the bay where the institute is, on the beach at low tide, when you flip over rocks, then sometimes you have a pile of these hermit crabs, like 100 of them, just sitting there. And then within like half an hour, you can collect, I don't know how many, as your grazing army, and can put them not with the really tiny babies, but for like slightly larger juvenile corals, these are also ideal. But yeah, you can't really get them in these quantities here.
Yeah, that would be a bit tricky. I guess that's one strength of the urchin, that you can spawn them. But yeah.
Yeah.
Would you ever want to try that?
What's that?
Would you ever want to try the urchins?
Yeah. Of course, that's definitely on the to-do list. But it's also one of the things that requires some time and preparation and all that. But yeah, definitely would love to do it.
I think that would be awesome to see as well.
Yeah.
Right. Next point was something that you've briefly mentioned. I kind of had to hold you back. I wanted to save that because that's probably one of my favorite topics out of them all. Is if we talk a bit about genetics and the hybrids and the chimeras and all that good stuff that you are in the unique position to even enable in this hobby. Because if we look in the ocean, hybrids, if we even know that they're a hybrid, right, are very rare. Chimeras come in even just as rare. Every now and then you find one, you see one, incredibly rare. Yet you breed corals, and it seems a lot more common, or something you can maybe even attempt to force to happen.
Yeah.
Let's break down the terms really quick. I thought maybe we should shed some light on what's what to start with. So what is a hybrid? What is a chimera?
So hybrid is a cross between two either different species or subspecies. Like they need to be genetically closely enough related so they can fertilize each other, and not just fertilize, but you have a full development, because sometimes fertilization can happen, but then the embryo doesn't develop properly and then it dies off, right, if it's not closely enough related. And funnily, in the acros, I really wouldn't want to be a coral geneticist, because especially not with the acros, because they're so complex and surprising, and we're probably going to end up with like 600 species in the near future, at least.
Currently, we're at 400 something. Probably more.
Yeah, by the end of that podcast, it's probably 500. I don't know.
Yeah, it's a lot. It's the largest genus of coral, right?
Yeah, and that's still in revision. There's so many, because there's so much regional speciation in the, especially in the Pacific. It seems like you got certain atoms when you thought originally that it's just say hyacinthus across the Western Pacific Islands, but then it turns out that you have regional populations that are genetically so different. Apparently, I don't know where to draw the line. I'm not a coral geneticist, but they seem to be so distinct that they should be their own species. And then you have one atoll where you have on a population on, let's say, the East Coast that should be its own species, and it's only been found there. So then also from a conservation aspect makes it quite interesting, because then you're one heat wave away from losing an entire species.
Yeah.
But that just as a side effect. So the hybrids are crosses between distinct populations that are still able to interbreed. And for acro, there's some species that are apparently very closely related genetically, although they look completely different, and that is apparently the case for Nyos and Echinata.
Oh, really?
That are within the same cluster. Of species.
They don't know, like Echinata is like a bottle brush shaped acro, and then
really
the Nyos is more like a cushion shape, a corimbos shaped one.
Yeah, it's pretty wild, and that's also when we got Fijian Nyos that was 2021, and the offspring from these, so they looked like more or less like tenuous, right? And then the offspring, morphologically, even though they grew up under the same conditions, same flow, same water, same light, same everything, some of them are more the classic shape, and some have these really long, thin corallites, like some of the ones I had at Interzoo.
Yeah.
That, and then also color-wise, they then look more like the Icefire Echinata,
yes,
but with a weird shape and still the long corallites, but they all come from the same parent pool. So the thought was, or is, or the hypothesis, let's put it like that, we don't have like genetic data on that yet, that the parents we got may have been, or may be hybrids, but morphologically you can't really see it yet. But if these hybrids are then crossed again, then some of the offspring will get more of the Echinata traits, some will get much more of the tenuous traits, and some will get the intermediate forms. And that's maybe why we have this wide variety in morphologies and that batch.
That's crazy. And that's something you even see in your finished corals, like in your baby corals.
Yeah, the one we have sitting here, or like the one that I had at Interzoo.
Yeah,
the ones. Yeah, that is especially that batch of the Fijian tenuous. Yeah, it's super fascinating.
How long does it take for these traits to be visible? Like at what point? How old does the coral have to be? Is that like at the half year or at the 1 year mark?
Yeah, between half a year, I would say half year to like 3/4 of a year. Like in that range, you can see that the morphologies are different, and that we have that in the Kentai as well. Not as crazy, at least so far. But there we also have some that have much more compact growth forms, and some have more elongated corallites. Even though they grow up under the same conditions. Right. Before I was thought like flow had a super strong impact on morphology. Of course, it does have an impact, a significant one. Of course, also if you see the corals growing on a reef flat or on the crest where you have like super strong currents, then they get much more stocky and compact than in areas like in a lagoon. But still, if you have them in the same conditions and they still have these crazy differences.
Yeah, that's cool. And until then, until you find out how they grow out, it's kind of like a mystery. It's kind of like you don't know what you're getting.
Total black box. Yeah, also color-wise, because the variability is it's pretty wild, especially in the Kentai. But I think color is a different topic. So now we have the hybrids.
Yes. Now what would be
drifting off again?
Yeah, it just happens, but it is fascinating.
Then the chimeras.
Yeah.
So chimeras are, I think originally the word comes from like a old Greek mythological beast, right? That consists of different animals. So a chimera is a colony in the case of coral that consists of several individuals, meaning that you have multiple larvae that settle closely to each other of the same species, and when they grow and touch each other when they're still young, then they're not fighting. Sometimes they are, but often enough they're not fighting, so that immune response that usually activates that fighting response is switched off, and then they fuse into one colony. And that has the advantage for them that they're already larger, right? They have different traits that could help them, but that's a point I can come back to in a second. And then they grow to be like morphology-wise a normal colony. But if these individual corals have different color traits, then you get that multicolored colony. And then there's also differences. Sometimes you have one that has one half is green, the other half is blue, or sometimes even with three, like green, cream, and blue. And then we have these spliced or swirled types where you have the mix of the different individuals going through an individual polyp. So you have a single corallite that has one side is purple, and the other side is green. But it, like shape-wise, it's a perfectly fine corallite, but it just has this or like a swirl of purple and a green. And if you would then take a genetic sample from the purple and from the green, you would find that it's two different two different individuals.
And that it's still in one corallite and one polyp.
Yeah.
So that's the crazy part. And of course, then that is highly desirable for the trade, for the market, because who wouldn't want a 5-colored Acropora?
Yeah, exactly. It's super fascinating. And now we're in the position where we want to look into that in a more targeted way. How to enforce it or create conditions that happens more often, because it's still we can produce them, but not in large quantities yet, because you can determine it a bit by how densely you settle the larvae, and larvae, they like to settle closely to each other. They can probably smell each other, and because it's an advantage to fuse early on, they tend to settle closely, I think. But if it's then too many, let's say more than 4 or 5, then we also had patches of like 50 or 100 that fused, and you're like, wow, that's going to be nuts. But they're never making it. They maybe a week, and then one is croaks and then takes the rest with them. But in nature, they're dispersed over so much more area, or so vast areas, that it's
very rare.
Generally fairly unlikely that several find their way that close to each other.
So could you try and force that to happen? I mean, I'm just thinking out loud. Can you just literally take a tweezer and like push the larvae around and move them together into one little clump? Or is that not as easy as it sounds?
Yeah, there's like towards mechanical impact, they're fairly they don't like that when they're small. It's also different time frames. Some species, I think they have a longer window to fuse, but then we have some, or my experience in like Millepora, the window for fusion. We also have fused Millepora, but they start to fight much earlier then.
So it's the same principle as when you say they fight, is the same principle as if I took any 2 different Millepora frags, like literally I break off a branch, I put them on one frag plug, they're not going to fuse. They're not going to, they're going to fight, or it's going to be 2 separate corals always.
Yeah.
But it's
also with it, because then the immune response, or they recognize, hey, that's not me, I'm going to make the other guy go away. By any means I have, so to speak.
So you have that very short window when they're tiny, where this is possible.
For some species, that window is a bit longer, but it's still limited. And then even in the Kentai, we have some. Recently, Marine posted on Instagram we call it the Fight Club corals. So these are chimeras, mostly, that consist of more than 2, and then 2 are fused and are doing fine, and then there's third one, and then you see like building a wall. When they try to overgrow each other, and then they build this weird wall-like shape, and it's kind of funny to see how they develop. So it doesn't always work then, or maybe the third one, or sometimes only 2 that do the wall that by the time they touched, they were already not able to fuse anymore.
So they essentially settle too far away, too far apart. Okay.
Yeah, potentially. Or they are genetically too different. So I don't know where the line is then drawn when they are compatible or not. So that's still something also to be determined. But yeah, it's super fascinating.
Oh, definitely. Like I just, when you think about it like that, you start thinking, oh, which cool combinations could you create? Why not mix a purple millie and a neon orange one, or I don't know.
Yeah.
All that cool stuff that we'll hopefully in the near future see coming out of Germany.
Yeah, hopefully. Yeah.
So currently, how far ahead are you? Currently, you're currently on generation F1 essentially, right?
Yeah, and we have the F1 already in the simulated systems.
Oh, right? So you do want to create an F2 where you spawn your spawn corals again.
Yeah, that's the goal, and also to do a more targeted breeding,
like a line breeding almost.
Yeah. I mean, whether you breed tulips or guinea pigs or cats or whatever organism or clownfish, for instance. I mean, it starts with the ability to produce them sexually, and then you can select the ones you intend to continue breeding with. And with the corals, there's so much unknown that I have no idea that if I take a blue kentai and a yellow one, do I get majority green ones? Most likely not, because there's so many color proteins and everything and factors involved that I really have no idea how that is passed on, like transgenerationally. So there's still a lot of trial and error going on, or in the future.
Yeah.
I mean, you never know immediately what you get, because then the colors come out after a couple months.
So every batch you essentially get is super exciting because you might get something completely new that no one's ever seen before.
Yeah, you never know what you get. It's always that black box until the color differentiates and so on. So yeah, that's also really fascinating about.
But then once you start like designing the corals, once you really get into choosing, oh, these have great colors, but these seem to be more resilient to pests or temperature, then you may be able to get something going in that regard as well. Where, I mean, I wouldn't say no to Acropora that are immune to Acropora flatworms. I don't think anybody would mind that. I don't know if that's something that's possible at all.
But that would be a selling argument for sure.
Oh, definitely.
Regarding I don't know if you could like flatworm immunity. I don't know if that. No, I don't know.
Well, maybe with a higher slime production, like looking at the green slimer is often considered very immune against flatworms.
Yeah. I would have to figure out what trait. But the way to go, and that's also I talked with other people about that as well, that if you have an established, let's say, a millepora that has been in culture and has been shown that this particular individual, maybe it's not like the most popping color, it's a brown one or whatever, but it's super hardy.
Yeah.
And it's not that susceptible to parasites and whatnot. And if you take then that and take a particularly beautiful one and breed with these to see maybe you get really beautiful ones that are at the same time more resilient. The resilience itself, is also something that we can see that it makes a difference whether or not they grow up from second one under aquarium conditions. Because the kentai compared to and we also work with imported parent colonies. That's such a difference. And I mean, I've known stores that said they stopped doing anything with tenuous or kentai because you can't make enough revenue with them because you have often so many losses that it's not really worth it. That we can offer the kentai that are quite a bit more forgiving and don't come with the usual mortality in the stores as well. So that's
I might know a thing or two about the mortality of tenuous kentai and Befaria as well, to the point where I don't think I want to even try them anymore. Because I out of all the Acropora, I think that specific one I don't know what it is about it. It doesn't work in my tank, but maybe a tank spawn one made in Germany, maybe that's worth a go.
Yeah, I think that's what we see also in fish, that they're more adapted, more easygoing, that you can like the Mandarin fish that you can feed with like dry food and stuff like that. So that's also in the freshwater aquaria, that fish that used to be super finicky are over time much more easygoing, and I think we have the same effect here. So I'm quite sure that these corals will make some people, or hopefully a lot of people, very happy.
Oh, definitely already. So one more thing about the whole genetics and the lineages is you've implemented something. Your little frag plugs, coral plugs. They have an ace up their sleeves too. They have an ace up their sleeves, a little party trick that helps track what coral it is, where it comes from, and all that good stuff.
Exactly. So we thought it would. So we have this little gap. I don't know. It's probably just audio only, right? So
we can see it.
Okay. Then there's the little gap, and then we have these NFC tags. And initially, we thought it would be great for us internally to keep an overview of the corals that are being produced. Also, in terms that over the year we can still know which coral is from which year, from which parents, which species, and so on. Then we thought chipping them would be a cool thing to do, but also for customers. That you can basically take your smartphone and hold it next to the we call it the ID stone, and then it shows you on the phone what species it is, when was the spawning, so you can have a little birthday party for your coral, and then attributes like where come the parents from and what kind of color did it have when it left our facility. And that is now being written onto the plug, and each coral is an individual. So that the kentai that are now the first corals to enter the market, they're all, we call them the unique coral, meaning these are all unique genetic individuals. That also means if they leave our door here, then we don't have them anymore. And the person who buys it is the only person in the world, basically, that owns that particular coral. So we thought it would be cool to also give them kind of a personal ID. And with all the information. And we're also working on our own app that will be released, and over time we will add functions, so like more information, but also things like sensitivities, what to look out for, like some care tips, what are the conditions. We have them in here. So the kentai, and some people are surprised, we have them at rather a bit higher nutrient levels. So phosphate, I would say usually around 0.1 to 0.15, or I sometimes go down to 0.07, but not below 0.05 usually. I mean, you can also keep them at lower nutrient levels, but generally I rather have a bit higher phosphate. And stuff like alkalinity, we have them at 7 to 7.5. Like light intensity at 2 to 300, but we also experiment with highlights, etc. But that information will then also be added. And one other function we work on is that you can basically start your little coral family album. Right, you scan the coral. It's being saved in the app, and then you can, for instance, take a picture when you bought it and save it, and then over time add pictures to see how it develops and so on. And I think that it's like adding a few more layers to the whole concept.
Yeah, every coral is unique, and now every unique coral can have its own story.
Yeah, exactly.
Oh, that's awesome.
And it's also kind of a safety or like a certificate of, how would I say, that it's an original from us. So we have the plus.
Yeah.
But if there is and of course, once they enter the market, they will probably be fragged and then people share them, which of course is fine. But what we would not like to see is that corals would be sold under our name because they are marketed under a brand name. So if there's vendors offering SciReef corals and they don't have a chip, then they don't come from our facility, basically. So that's also kind of a reassurance that without a chip, they're not directly from here.
Well, lineage tracking, I think, in general, is just very tricky in this hobby. Where, well, once I have the coral, I have got the chip with it, I give the coral to someone else, a frag of it, part of it. Now, that person grows it out, gives it away. At some point, it gets a bit sketchy. But at least down the line, if you follow who got it from who, if you follow down the line, there should be someone with the original NFC chip that got it from you.
Yeah. So the first two steps, then we can be sure is, but once they are in the hobby, and I mean it's.
No, but it's something really
understandable. I mean, they're like price-wise, it's still quite a different category than.
But it's something to be proud of as well, because if the coral is unique, you're the only person that has it, and I think we talked about it a bit. It's kind of a bit of a random factor as well, what the coral will turn into. I mean, I think by the time you get it, you kind of know what you're getting, but you never know until you grow it out. And
exactly.
Then it's even more rewarding because you're the only person in the world that has this one coral.
Yeah, exactly. But of course, then you can still share it, or if people, I don't know, 2 or 3 people say, "Hey, let's get one together." That's fine too. But the thing is that like we formulate that in a we don't want to have knockoff SciReef corals like in a officially being sold or something like that. So like the first 2 steps
I see your concern.
Are kind of like with a certificate, but what happens beyond that is not in our hands anymore. And that's also of course the challenge that we always have to work to produce new stuff, because you can't, I mean, for the next years always bring out the same kind of coral. Of course, at some point, then it's not a novelty anymore. And there's so many more species to work with, and especially to explore things like the chimeras and targeted, targeted breeding in terms of color, morphology, and all that. So that, all these complex processes and the diversity, of course, that makes me very positive that there will always be new stuff coming.
See, that almost gives me a bit of FOMO, a bit of fear of missing out, because when you say you always try different combinations, different mother colonies, it's not always going to be the same one. It's like, what are the chances that the ones you just released the other day or last week, a week ago, were the first ones that entered the market?
I think the first delivery was about 2 weeks ago almost.
Right.
Yeah.
So what are the chances of those ones ever, like, of another batch of those, essentially?
Very, very small.
Because even if you wanted to, I guess it would be very tricky at this stage.
Yeah, like I think every year, and that's also why we always put in the year or spawning month to kind of like in wine culture, that you have certain years that every year is different, and that's going to be the same with our corals as well. And also, you can't really predict how many there will be. If you have a good run and everything goes perfectly fine, or as fine as it can, then you can potentially produce quite a lot. But there's certainly going to be spawnings on the same species where you don't have high yields, and then you have very limited, or even more limited as now, amount of corals, and then from that year maybe we can release only that number.
See, I haven't thought about that at all until just now, which makes the whole thing even more awesome, if you ask me. So that's actually pretty cool. So every year you get, I say every year, I guess it takes about a year for them to grow up. That's why I said that. But I guess you could have multiple releases a year, depending on how you scale your operation.
Also what we do. We because some species take a bit longer to, like the Millepora grows very horizontally at first. They take a bit longer until they're turning into a really nice coral. Also, the color is a bit different in them. So first they're like neon green, like the ones I had at Interzoo.
Yes, almost a bit glittery as well.
Yeah, they're all green, like neon green. Then they turn brown. And then they, at some point, bring out coloration. So they always take a bit longer. So right now, we're also working on new species. And then the second concept, I said the Kentai that we released now are the so-called unique corals, meaning every single coral is genetically unique. And then we'll also have the so-called twin corals that are also spawned here in Wilhelmshaven, grown from an egg. But these are then the really fast-growing species like the Austera. And these, at a certain size, we will then asexually basically cut up. And then there is not a unique genotype, but that's why we call them twins, because then you're not the only person who has it. But on the other hand, these are then also much more affordable than the ones that enter the market now. But so we always then have and that's also on the chip. It always says whether it's a unique coral or a twin coral, and these are then the two basic categories.
Awesome. So on the market, we said they've been on the market for a whopping 2 weeks at the time of us recording this. So what's currently on the menu and where can we get it?
So right now we have the Acropora kentai, the unique type from last November spawning. And we categorized them into 4 different grades because it was a bit tricky to come up with a system for naming. Because if you look closely, it's really hard to find 2 that look the same, even if you have a tank full of them. So they all have their little differences. And some color varieties are much more popping and some are more rare. And then also morphology played into it. Some have this really, really cool symmetric, like a tiny perfect colony shape, and some are not as symmetric, which doesn't mean that they won't be, but at the time right now they're not. So we have the grade 1, which is our really nice standard coral. So the grade 1, they all have really nice colors, but maybe not the most popping ones. And not all of them have that really perfect bonsai shape. But still, they're all unique genotypes, and the shape and color is at the moment now that they leave our facility. So I'm sure, they will turn into absolutely fabulous corals. And even now, if you look at them, we have a lot of pictures online, so feel free to check them out of the different grades. And then grade 2 is the ones that either have really popping colors or a really beautiful shape, like the nice compact, or depending on the morphology, nice colony morphology. And then grade 3 combines both. So they all have really striking colors, and at the same time a really beautiful colony shape. And then we have the grade 4, and these are the
highest
chimeras.
grade. Yeah. So that's the creme de la creme, the best of the best.
Yeah, and the ones of which we really don't have that many. So there's only a few that have now entered the market, and we'll see how we proceed with these. But these are now the 4 that are 4 categories that are available.
Yeah.
And fittingly, at the moment, it's also 4 vendors, all based here in Germany, where you can get them. Although, as of right now, it's still 3 Claude from. So let's name them first. It's Fauna Marin, it's Coralixy, Aqua Design in Oldenburg, and Broodstock, Meeres Aquaristik, based in Bremen. Coralixy, Aqua Design, Broodstock already have them. Broodstock got theirs, I think, Tuesday, 2 weeks ago. Claude was still in China until end of last week, and he wanted to be there when they arrive. So
I can see why.
His will ship out tomorrow. So
awesome.
Wednesday, he should get his. So these are the 4 vendors at the moment, but we'll also expand that vendor network. So initially, we chose vendors that we've all known for a really long time. And we know the corals are in good hands, and they have the competence, and also to the right customer network. So they're really established stores, and we have a very good relationship with them. And now, step by step, we will expand the network. But so in the beginning, also logistically, because we have to run the place here. We have to do a lot of administrative stuff, then you have to communicate with everyone. So if we would start with like 20 vendors, that would be like challenging.
Well, it's something special for sure. So those 4, do they ship just within Germany, or is it EU-wide?
EU-wide.
EU-wide, okay. So the whole EU...
As far as I know, Aqua Design is setting up their online store, I think, as we speak. The other ones already are shipping EU-wide, and the corals are also what you see is what you get. So since they all look different, the stores they all have different. Even if it's the same categories, they all still look different.
So they all take their own pictures, is what you're saying?
Yeah.
And they're all unique anyways to begin with, so...
Yeah. As far as I know, every individual coral is shown then online.
Are there any plans to expand to the US? Because I'm sure people are going to ask.
Yeah, there are. So we're already in contact, and of course we don't want to let our friends across the pond wait for too long.
No, I know there are some collectors just waiting for this opportunity to come up. That's for sure. Yeah. So how long will they have to wait? What's like the rough time frame we can expect?
I mean, we'll start with the paperwork because we have to apply for the CITES because it goes outside EU, and it depends on how quick the authorities are. So the German export, I don't think will take too long. I don't know how long the Fish and Wildlife Service processing time will be, and then finalizing all the logistics. So
what's the goal? The rough goal? What's your goal for US export?
A rough goal would be sometime in the let's say second quarter next year.
Awesome.
That would be perfect. Would be good, I think.
But it's coming to the US as well, so because I know a lot of people listening in or watching are gonna be from the US, and they definitely want to know.
Cool.
And so the species on the menu again, that was the tenuous right now. The
Kentai.
The Kentai.
Yeah, and then shortly after the Austera.
But they're not out yet, right?
Not yet. No,
not yet. Okay. Soon.
Yeah.
Okay.
Soon. Very soon. And then we're working on the Nasuta, and then the Fijian tenuous, Millepora, Microclados, and then there's a few more in the pipeline. So hopefully,
very good.
Regular intervals we can bring something new.
Yeah, perfect. Can't wait.
And then we, of course, want to branch out into different types of corals, but I can't make any concrete estimations.
TBD. That's fine.
Yeah, exactly.
TBD.
Yeah.
Let's see what's left. What are some lessons that you've learned in your endeavors from you starting out till right now? What have you learned that changed your view on Acropora or on reef keeping?
Yeah, one fundamental thing is because we work a lot in the dark, figuratively and literally, that looking at the corals at night, especially the acros, is absolutely. You can learn a lot from that, how they react at night, how far they're open, if they're in feeding mode, because sometimes even if they're sort of open during the day, but don't open up at night, then something may be wrong. Doesn't have to be, but it's always good. So if I see a coral really super wide open at night, then
I actually agree with that. Yeah. I also like to think that an Acropora that opens really wide at night, even if it doesn't have the best color yet, or it's maybe a bit dormant, it doesn't grow too much, but it's open at night, that's usually a good sign.
Yeah, exactly. Yeah, that was one thing. And then, sedimentation is also, for the baby corals, it's kind of obvious that it's a problem for them. If they're being smothered and they have to produce more mucus to get it rid of the sediment, and you get bacterial growth. But in larger colonies, even if you have a lot of changing flow and turbulent flow, you always have some areas where the flow isn't going, or that are kind of stagnant and stuff is accumulating. And in nature you have the changing tides, and then sometimes a storm comes rolling through, but even there you see often patches where sediment and they start to lose tissue. So a while ago I started to set up Förderpumpe.
Return pump,
that's the word. Yeah, a little return pump in each system with a long silicone tubing.
Yeah.
And then ideally once a day we take the silicone tubing and basically shower our corals. And sometimes, even if you can't see too much sediment, it can be surprising the amount of stuff that comes flying out. So, and since showering has been proven beneficial for humans, I think it's for corals, it's also a good thing. And especially in species like Austera, that is super hardy and super fast growing. The one thing it really doesn't look to like is if it has sediment accumulating, and then you often quickly get issues there. But ever since we do the coral showers, that has basically vanished. So that was also one takeaway from working with babies and then thinking more about like sedimentation and so on.
Yeah, I think, especially also if you have larger colonies, you always see a bit of die-off in the middle. Could be from light, could be from flow, but especially with like tabling corals, you might want to make sure that nothing settles on or in them.
Yeah.
So, exactly. No, that's a good one. Definitely.
Yeah. And then just to try it out, even if you can't see that much lying or in there, sometimes you would direct the flow right into it and see all the particles come out.
All right. So to wrap this up, what is your future outlook and your vision for coral breeding and this hobby in general?
I think this type of breeding operation as we do, or that methodology will be an essential part. Fragmentation will definitely always be a relevant method to produce corals, either for the trade or for restoration. That's no doubt about it. But to be more independent of the supply chains, and especially also the aquarium adapted corals, like from the aquarium for the aquarium, is often what we say. I think that's going to play a big role. And the creation of new interesting corals, because also quite a few vendors we talked to, they said, you always now and then you get new color morphs or something, but generally there's a lot of the same on the market, and it has been kind of the same level, well, maybe not level, but it has been a very similar development in the last, let's say, 10 years. Technologically, we've advanced a lot, and fish breeding and invert breeding has made huge steps forward. But for the coral culture, it was, on a similar level. And now I think we can give some impulse and also bring more of that or more depth to the fascination about corals, because now, since we now can close the life cycle, then that makes it gives the animal itself more depth and more complexity and more to think about, and also where they come from and what may be possible in the future. So I hope that brings in some,
definitely. Yeah, especially some imported corals. You wouldn't, as the end user, as the aquarist, you don't really know where it comes from. You don't really know where in the ocean it comes from, what farm it comes from. It's all not super transparent for the end user. But now with your corals, that's a bit different.
Yeah, what you may bring in with it, like with the imported corals, sometimes surprising the fauna that is brought in with it. So,
definitely can be surprising. That's for sure.
So that is an issue then that is not relevant anymore.
Let's see. What would be a coral you would absolutely love to be a part of your project, a coral that you would love to spawn and offer to the market?
Definitely the LPS, like the Trapeze and the Scholumia, but also like Cinerina. I think this is absolutely beautiful coral. Cinerina, I mean the Cantophilia, take quite a few of the LPS. I like a lot. I mean the Trapephilia are my favorites. Marine's favorites are the Cinerina.
Yeah.
So I think these are on the to-do list as well.
Yeah, if you could spawn some master grade Scolies or something like that, I mean, people wouldn't be all too mad about that.
Yeah, I think so. Yeah, would be cool to see if they can also fuse to do like some funky chimeric Scolies.
I mean, they seem to naturally even show that tendency, so that would be very interesting indeed.
Yeah, it would also be interesting to see if like the really the huge color differences to do genetic testing, just to see whether maybe they're actually fusing early on. Who knows? There's still so much unknown.
Yeah.
Yeah.
Which do you think we would ever see some corals from parts in the world that aren't even accessible for the trade right now? Let's say, for example, like Caribbean stony corals, like the elkhorn coral, stuff like that. Would that ever be feasible or even possible?
For the elkhorn, I doubt it, because I think they're even Appendix 1.
That means for
the CITES that are even more strictly regulated, or at least they're in the Endangered Species Act, so that's even more strictly. But I mean, not maybe through F2 generation for these cases, but in other cases, I can't imagine that if they're not being actively wild collected for the purpose of then entering the trade directly, that maybe there's once that possibility is commercially available to reproduce them in that way, that there may be a regulation or possibilities to do that.
No, it would be good if the aquarium industry would be, well, not just seen in like a bad light that we're harming the ocean. No, we're actually supporting the ocean. We're giving back to the ocean. We're raising awareness in probably the best way possible.
Yeah, exactly. Yeah, I think that's also sometimes wrong. Like the great if it's being portrayed as like just taking stuff and consuming. But I mean, if somebody like us has such a deep fascination for the coral and for the whole ecosystem, that you go to all the length, like financially and also like the time and effort you have to put in to put an actual reef in your in your home, to then say that person is just exploiting the ocean, that's not really a good fit, I would say. So that's not the right way to put it. I mean, there's definitely things that can be optimized or have to be optimized. I think it can be done in a way that everyone benefits from it.
Right. I think one last question we can sneak in, and I think then we should end it. What is like the last like biggest problem that you're still facing, that you're still trying to solve, and maybe something where science could help, other hobbyists can help? Like, what is still a big problem at hand that you haven't been able to solve?
We don't have one really substantial problem that we categorically can't solve, but often unpredictability, that you can't really predict, okay, how well is that spawn now gonna go, because there's so many tiny little
like so many variables.
so many variables and mechanisms that it's often hard to predict how one particular one is going to go. Yeah, one thing that would also be great for hobby and research of these two worlds of like coral science and coral hobby, if there would be more overlap. Because often these are very different bubbles and they each have different approaches to solve problems, and it would be very beneficial or a lot of synergy if there would be more overlap. Because often, like in the coral research and altering corals over the long term in an aquarium is often a very large hassle and an issue.
I mean, it's I dare say almost a difficult skill to acquire to keep a reef tank. It's a science on its own.
Yeah, definitely, and that is often not acknowledged by the actual scientists. If you will, they don't. Yes, you don't learn that at university. Right?
Yeah.
You learn it by spending tens of thousands of hours of reading stuff and looking at coral, right, and doing all sorts of things and trying it out. But you can't really learn how to run a reef tank. There's no university program for that. So that if they don't bring that skill, or if you want to work in that research field and you don't bring that skill with you, then you're not gonna learn it.
There's a lot of trial and error involved anyways, and if you work with, I don't know, endangered species, that might be a costly mistake that is just waiting to happen. I think I've heard something similar, and I very much agree with it. And it was every scuba diver, like everybody who likes to go diving in the ocean, should have a reef tank. And everyone who has a reef tank should go scuba diving in the ocean. So you kind of see both ends of the spectrum. Because you can learn so much from the other and apply that knowledge. Or if you go diving and you know what fish you're looking at, you know what corals you're looking at, you know what critters are lurking around. And the other way around is like, if you know what corals look like in the ocean, and now you see them as a very tiny fragment of them in your own tank, I think it's just that what you described is that overlap should definitely be used or made use of, because it's just brilliant.
Yeah, it should be pushed for sure.
But I guess that's also what we're doing here, so people can learn about coral spawning. People now know that you provide tank spawn corals made in Germany. And it's just absolutely exciting what you're doing. And
Thank you so much.
Essentially what the aquarium industry has unlocked thanks to you. So,
not just me, of course.
Well, you're here talking to me, so you get to you get the credits for now. No, so that's awesome. Yeah, and I think I'm not the only one excited to see what the future holds for our industry, our hobby, and SciReef, of course.
Yeah, exciting times for sure. Yeah, thanks so much for having me on.
Yeah, so where can people learn more about you? Where can people find you? Facebook, Instagram?
Yeah, mainly Instagram is where we are most active, and that's where we're most regularly posting things. Also, then also YouTube. There is on the Tropic Marine channel. There's a lot of videos about our research projects at the university and also about SciReef.
Awesome. So if people have any input or questions now, leave it in the comments and I'll make sure Sam answers them.
Yes, sir.
And I'll try my best as well, but I will refer to you if it's something more technical.
Yep.
And I think that was a very long episode now. Thank you so much for being part of it. And
it was a pleasure. It was fun. Chatting about corals and coral spawning is always a good time.
Awesome.
Thanks again, and good night. And to all of you out there, take care and happy reefing.